Good afternoon. We are Darío Delgado, Adriana Barrete, and Alfredo Rivera, and today we will present you the Biotechnology Applications Network focusing on base editing applied to T-cell acute lymphoblastic leukemia.
As a practical application of red biotechnology, we have selected a groundbreaking case published by the BBC that is titled Pioneering New Treatment Reverses Incurable Blood Cancer in Some Patients. But what happened? In this selected case, scientists used a revolutionary technology that is called base editing to treat an aggressive incurable blood cancer. By applying precise genetic modifications to donor T cells, they transformed them into a living drug that is engineered to destroy cancer cells without attacking the patient's healthy tissue. The research was led by Professor Wassem Kassim and teams from University College London and the Great Ormond Street Hospital. The patient in the case is Alisa Tapley, a teenager who became the first person in the world to undergo this therapy and remains cancer-free today as a testament of this success. The trials took place in London, UK, while Alisa's journey began in 2022 and currently her cancer is undetectable. Now, 8 more children and 2 adults with T-cell acute lymphoblastic leukemia have been treated. The broader clinical results show a remarkable 64% remission rate. Why is it noteworthy? It's because it marks the transition from science fiction to clinical reality. Unlike traditional CRISPR, the base editing allows for the alteration of a single molecular letter in the DNA, providing a last resort hope for terminal patients. But what is red biotechnology? The red biotechnology is the branch of biotechnology that is focused on medicine and human health. It involves using living organisms to develop products or techniques that help prevent, diagnose, or cure diseases. Its name comes from the color of blood, and not to its focus on health and human life. Red biotechnology covers all uses of biotechnology in healthcare, including the production of vaccines and antibiotics, the development of new drugs, introduction of molecular diagnostic techniques, regenerative therapies, etc. This branch allows the creation of personalized treatments based on an individual's genetic profile, such as the base editing seen in the leukemia trials. Also is the backbone of modern drug development, enabling the production of recombinant proteins, like insulin. Utilizing tools like CRISPR, the red biotechnology provides the potential to cure previously incurable genetic disorders and aggressive cancers. The case we have selected is an example of regenerative medicine. But why exactly does it fit into this category? The first point is the restoration of immune function. Since regenerative medicine aims to restore or establish normal function in damaged tissues or organs, in this specific therapy, the patient's entire malfunctioning immune system is dismantled and subsequently regrown. This is achieved through a combination of gene-edited cells and a follow-up bone marrow transplant, effectively rebooting the body's biological defenses. Another core pillar is the use of living cells as a therapeutic medium. Instead of using a traditional chemical drug, the doctors are engineering biological units, specifically in this case the T cell, to act as autonomous defenders. This is what we call the living drugs. And finally, the use of the base editing to rewrite the instruction manual of the human cells is a hallmark of advanced regenerative techniques. By modifying donor cells to become invisible to chemotherapy and highly specific cancer, scientists are not just treating symptoms, are creating a tool to repair a fundamental systematic failure in the blood.
Now, let's talk about the science behind this amazing case and who actually benefits from it. In this case, doctors didn't use traditional surgery, just hearing aid. They used something called base editing. You can think base editing like it's like a molecular pencil, but most genes editing tools act like scissors that cut the DNA, which can be a little bit risky, but base editing is much softer than just erase one wrong letter in the genetic code and write the correct one. In Aisha body, there's a tiny mistake in a gene called OTOF. This mistake stopped sound signals from reaching her brain, By using a harmless virus to carry the pencil into the ear, the doctors correct that single letter. The results were mind blowing. In just a few weeks, Aisha went from total silence to hearing almost normally, but she can now hear someone whispering, which is a huge scientific win. This shows that we are moving from just treating symptoms to actually curing the biological cause of deafness. Now, let's talk about the science behind this amazing case and who actually benefits from it. In this case, doctors didn't use traditional surgery, just hearing aid. They used something called base editing. You can think base editing like it's like a molecular pencil, but most genes editing tools act like scissors that cut the DNA, which can be a little bit risky, but base editing is much softer than just erase one wrong letter in the genetic code and write the correct one. In Aisha body, there's a tiny mistake in a gene called OTOF. This mistake stopped sound signals from reaching her brain. By using a harmless virus to carry the pencil into the ear, the doctors correct that single letter. The results were mind blowing. In just a few weeks, Aisha went from total silence to hearing almost normally, but she can now hear someone whispering, which is a huge scientific win. This shows that we are moving from just treating symptoms to actually curing the biological cause of deafness.
In this slide, I will discuss the main ethical and regulatory challenges of this treatment, known as the 3.2 million drug. First, unequal access. We all know that not all of the families can afford the most expensive treatments in the world, which creates this risk that only people, rich people in countries with high level insurance will have access to the cure. So this brings us to the debate and the questions on distributive justice, because is it right to allocate so many resources to a single patient when we could benefit thousands more? The second, the expectations versus the clinical reality. Many parents accept the treatment driven by hope, and even though initial data did not have clear improvements in all children, here informed consent is a key. And finally, third, genetic privacy, as it involves long term monitoring of biometric data and DNA in minors, raising important questions about protection and the use of that information. And well, yeah. Now, let's analyze the safety and risks from a regulatory perspective. The treatment was approved by the FDA through an accelerated process based on a biological marker rather than definitive clinical evidence, raising questions about whether it was scientifically sufficient enough. In addition, there is a social pressure in rare and deadly diseases. Should regulators be more flexible or remain strict? In terms of risk, there is a long term uncertainty about the duration of the effect and possible side effects, and the body could reject this viral vector, preventing a second dose. So, in conclusion, we are entering in a new era of genetic engineering with increasingly precise editing, almost letter by letter. Base editing is expanding the treatment of these diseases previously considered not with cure, not only leukemia but also other cancers and genetic disorders. In addition, there are plans to develop edited and stored T cells ready for use in different patients. So the future is promising, but it must advance alongside regulation, ethics, and responsibility to ensure fair and safe benefits to all of our society. However, along with the opportunities, there are also obstacles. One of the main risks is of target editing, which can lead to unwanted mutations in the long term. Furthermore, it is a complex technology that is currently limited to the specialized centers. Then the social acceptance is likely to increase, but the big challenge here is to ensure equitable access and economic sustainability. Ultimately, the future of the genetic engineering depends not only on science and also depends on how we as a society decide to apply it.